Okay, thank you so much for this kind invitation and also a possibility to present my work even in 10 minutes. So let's try, but it will be different from the previous talk. But introduction has been done at the morning in the same session about protein-protein interactions. So what we are here. So how you do? So. Okay, so first of all, what is the question of my short presentation and my work? So the question is how evolution produced a new protein function, particularly for stress response proteins. And to address this question, we developed a database and a tool that called pastoral protein-protein interactions of stress response genes in subterranean and fossorial animals. So for this question, we collected three subgroups of animals living above ground, subterranean living underground, and fossorial. This means they sometimes above ground and sometimes underground, and or they build some holes and utilize both ecologies. So for this, we also used all these tools, particularly KEGG orthologs, to combine different protein families according to the orthologs from these organisms, all three groups of organisms. And then using protein-protein interaction database BioGrid, we utilized our server called GP, chimeric protein-protein interaction server, to rebuild protein-protein interactions for all protein family in order to understand how stress response genes produced in evolution. So just briefly, I would like to explain how we look on protein-protein interactions. So we take a BioGrid for every protein, and to rebuild, in the case if we don't have protein-protein interactions, we need to rebuild all this network. To rebuild all this network, first of all, we pre-calculated for known proteins, protein-protein interaction network, and used domain-domain co-occurrence. So any protein in the network can be presented using building blocks of protein domains, right? And then using protein domains, if they appear in the same network and produce protein-protein interaction, so they can be calculated in domain-domain co-occurrence table, right? And then using log odds, we can calculate observed probability versus expected probability to be in the same network. In this way, we, after calculating this domain-domain co-occurrence, for every possible protein, novel protein, in unknown protein network, we can rebuild protein-protein interactors using this GP server, GP server. So as I already said, we took different types of organisms, particularly living always under above ground, like human, mice, and so on, insects, and animals living underground, like these animals which never go above ground, and they have particularly rebuilt all the hypoxia, oxidative stress, and circadian clock networks. So how they did this is exactly what we would like to study. Let us look first of all on all these animals in our groups and try to use only multiple sequence alignment, only sequence characteristics of the protein, particularly for a pass domain. This is a domain participate in stress response proteins. And if we try to rebuild using only multiple sequence alignment, we receive just a normal phylogenetic tree, but produce evolutionary correspondence distance between these animals, just evolutionary distance according to time of development from previous ancestors. So no connection to the ecology, no any connection to an ecology. You can see fossorial, F, S, and above ground, they all appear without any clusters. But now let us look on protein-protein interactions as the base for building this phylogenetic tree. Protein-protein interaction and not multiple sequence alignment, not just sequence information, but functional information from protein networks. In this case, for hypoxia genes, we clearly see clusters for fossorial, for above ground and subterranean animals. This means that clustering based on protein-protein interactions reflect shared ecology between the organisms. So the function is important to build ecology, not just a sequence information. And in same way, for additional stress like heat shock, we can rebuild clusters for ecology using protein-protein interaction networks, as well as for circadian clock. So these animals build systems as a response using protein-protein interaction networks for these protein families. In addition, we looked also on composition of disordered domains, and particularly on codon usage preferences in ordered domains, as I presented you in CHIPI, and disordered domains to understand the evolutionary connection of codon usage preferences. So if we consider non-optimal codons, this means codons that utilize wobbling, they are non-optimal. And codons which are perfectly matched to tRNA, they are optimal. In this case, we found that subterranean organisms, they use more non-optimal codons, and these regions correspond to disorganized or disordered domains. So significantly more disordered domains have been found in subterranean organisms as a response to the stress, particularly hypoxia stress. And now the model of evolution to recognize all these building blocks of protein domains, evolution can utilize to build it in two ways. One way is using mutations, point mutations, and another way is to take big parts of the genome and move it from one part to another, particularly using changes in protein domains. So we tried to rebuild all these changes in protein domains during evolution in these animals, in the same groups of animals living underground, above ground, and using both ecologies. So for this, as again, we took same groups of animals and collected more, even viruses and so on, to look again on protein domains using PFAM and orthologs using KEGG. And what we wanted to see, that protein domains can be moved full from one protein family to another, building novel protein families or novel protein function, like chromosomal translocations, similarly to chromosomal translocations. So, and indeed, we found more than 290 possible chromosomal translocations that build novel protein families. Here I provide an example for hypoxia proteins, which use PAS domain. This PAS domain moved from another protein family to using chromosomal translocation. Because PAS domain is so important for stress response genes, we looked explicitly on this domain. And then to understand why chromosome translocations can be involved, we looked on high-C data, and in this way, we found that indeed more than 60% in human and mice and 20% of chromosomal translocations, changes of these domains correspond to high-C contacts. So this is exactly, and this I would like to thank funding and my students participating in this work, Gon Karmi, Alexander Gorokhovsky, and Rajesh Chitroy. Thank you very much.